Randomized controlled trials, or RCTs, are studies that work out how effective a given treatment or intervention is in comparison to another treatment, or those awaiting treatment. RCTs are considered the gold standard for determining the ability of a given treatment to bring about the desired effects under ideal conditions. This is known as efficacy, as well as the safety of the treatment. Because of their strict steps and scientific rigor, they are regarded as being the highest quality of evidence for a single study, as shown in the so-called hierarchy of evidence. First, a carefully thought-out research question that tests the treatment. Ideally, it points to a possible scientific explanation or aims to solve an unmet need in science. Second, participants with clearly defined characteristics, for example a specific issue or health condition, are invited to the study to rule out any other influences on the outcome of interest. Ideally, a large enough number of people interested in the study are informed about the study and possible risks. It needs to be a large enough number to accurately represent, or be generalizable to the population of interest being tested. After receiving informed consent, the participants are then randomly placed into the treatment group and control group. A simple example of randomization is flipping a coin or rolling a dice, but most of the time a computer program is used. Randomization aims to ensure that both groups are ideally very similar regarding the characteristics that might be obvious or visible to the researchers and those that are perhaps less so. This ensures that the changes seen between the groups are only due to the impact of the treatment or so-called effect. The treatment group will receive the treatment being tested. The control group or control arm is used to compare and work out the more reliable impact of the treatment. The control group can take on many forms. For instance, it might be an active existing treatment. Standard care, meaning the usual care provided. Placebo, an inactive substance like a starch tablet, which looks exactly like the treatment being tested. Waiting list controls. This is when participants are put on a waiting list and practically do nothing. There is also such a thing as dose-response control, where controls have different doses or levels of treatment compared to those receiving the tested treatment. The next step is to reduce bias towards favoring the tested treatment by blinding. Single blinding means that participants do not know whether they are in the treatment or control group. In some studies, the investigators are also blinded to the participants' group assignment, also known as double blinding. Outcome measures. Outcomes are the things monitored during the study to see if the presence-absence of the treatment has an impact on those taking part. Outcome measures should be relevant, preferably measured the same way for everyone or standardized, and widely accepted in the scientific community. Analysis. Statistical calculations using suitable tools allow us to work out whether the treatment was indeed more or less effective, that is, whether the results were not just due to chance. Reporting. RCTs should be registered to reduce reporting biases, and reporting should follow standards like the Consolidated Standards of Reporting Trials or consort. So, let's apply what we learned so far with an example. Back to our model. Imagine a pharmaceutical company has developed a new drug, drug A, to reduce blood pressure in patients with hypertension, that is, high blood pressure. Study design will be a parallel group. The study involves 200 patients diagnosed with hypertension. Randomization. Participants are randomly assigned to two groups. Treatment Group A, 100. Patients receive drug A at a particular dosage. And Control Group, Group B, 100. Patients receive a so-called placebo, basically a sugar pill that looks convincingly like drug A being tested. Blinding. The study is double-blinded, meaning neither the patients nor the researchers know who is receiving the tested drug or the placebo. The trial lasts for 8 weeks. Blood pressure readings are taken at the beginning of the study and the end of each week. After crunching the data, the researchers will find out whether drug A is more effective in reducing blood pressure than the placebo sugar pill. In reality, RCTs are of course more complex and rigorous than explained here. Like any other study design type, an RCT needs to be interpreted carefully. To sum up, RCTs are very important for evidence-based care. However, their results should be interpreted cautiously, not as proven facts. Thanks for joining us. We hope this has given you a bigger picture of RCT. You can find a critique of RCT in part 2 of this series.